Welcome back to the deep dive. You know, it's fascinating to think about all the steps that go into making those little pills or capsules we take when we're not feeling well.
Yeah, exactly.
I mean, it's more than just coming up with a chemical formula, right?
Right.
There's a whole hidden world of processes that ensure that what we're swallowing is pure and effective. And today, we're going deep into that world, exploring the techniques used to purify and isolate drug substances. It's a pretty crucial part of pharmaceutical science, making sure that the medicine that ends up helping you is precisely what it's supposed to be.
It really is. It's like, imagine you're baking a cake, right? You need all the right ingredients, but you also need to make sure they're pure. You wouldn't want any eggshells in your batter.
Absolutely not.
In the same way, scientists need to make sure the drug substance is pure and free from any impurities that might be harmful or affect how it works.
And we're lucky enough to have access to some great material that goes into the nitty-gritty of how this is done. We'll be looking at some of the fundamental purification methods, crystallization, filtration, and chromatography, and we'll even see how these are scaled up to produce medications on a large scale. And don't worry, we'll try our best to keep it clear and straightforward, no overly technical jargon today.
We'll keep it nice and accessible.
Okay, so let's start with the basics. What are the core methods scientists use to purify drug substances?
Well, one of the most fundamental techniques is crystallization. It's a pretty cool process that relies on a substance's inherent property to form highly ordered, pure crystals under specific conditions.
So you're saying it's all about setting the stage for these drug molecules to basically want to solidify in their purest form.
Exactly. Think of it like this. You know how you can make rock candy? You dissolve a bunch of sugar in water, and as the water evaporates and the solution cools, the sugar molecules start arranging themselves into those beautiful, pure crystals. Drug substances can behave in a similar way.
So it's like those science fair projects where you grow crystals from a solution.
Kind of. But on a much more sophisticated level, of course, what's really important here is controlling the environment very carefully. Factors like what kind of solvent you use, the temperature of the solution, and the concentration of the drug all play a huge role in how those crystals form their size, their shape, and most importantly, how pure they are.
So you're essentially tricking the drug molecules to organize themselves into this perfect, pure structure while leaving all the junk behind.
That's a great way to put it. Impurities might not fit into that crystal structure or might have different solubilities, so they get left behind in the solution. Pretty neat, huh?
Yeah, that's really clever. So you're basically using the drug molecule's own natural tendencies to help you purify it.
Exactly.
Okay, so crystallization is one key method. What's another technique in the purification toolbox?
Well, another essential method is filtration. This is a more straightforward physical separation technique.
Okay, so this one's a bit more direct.
You could say that. Think of it like straining pasta after it's cooked. You use a strainer to separate the solid pasta from the water.
Yeah, I got that.
Well, in pharmaceutical terms, filtration is used in a similar way, but on a much smaller scale. It's used to remove solid impurities from a liquid that contains the drug substance.
Ah, so kind of like a microscopic sieve to catch all the unwanted bits.
That's a good analogy. The filters are designed with super precise pore sizes, tiny little holes, to trap particles of a specific size.
So it's like a custom-made net to catch only the impurities.
Exactly. And sometimes it can be used to collect the actual purified drug substance itself, especially if it's in a solid form after a process like crystallization.
I see. So filtration can work on both sides, removing bad stuff or collecting the good stuff.
Exactly.
Okay, got it. We've got crystallization for getting pure solids and filtration for removing solids. Now, the third method you mentioned was chromatography, which always sounded kind of mysterious to me.
It might sound a bit more complex, but the idea is pretty cool. It exploits the fact that different molecules have different properties, and we can use those differences to separate them.
So it's like sorting things based on their personalities.
You could think of it that way. Imagine a crowded race, and all the runners have different strengths and weaknesses. Some are fast, some are slow, some are better at navigating obstacles. Chromatography is like setting up a race course with different challenges so you can separate out the runners based on their abilities.
So you create an environment where the molecules have to kind of compete and separate themselves based on how they interact with it.
Exactly. There are typically two main parts to this race course. You've got the stationary phase, which is like the track itself, and the mobile phase, which is like the wind or the current that carries the runners along.
Okay, I'm following you.
Now, depending on their properties, some molecules are more attracted to that stationary phase and kind of stick to it, while others zip right through.
So some are like marathon runners who pace themselves, and others are sprinters trying to get ahead.
That's a great way to think about it. And this difference in how they interact with the system is what ultimately separates them.
And you mentioned there are different types of chromatography.
Oh, yeah, there are quite a few. Column chromatography is a common one where the stationary phase is packed into a column and you pour the mixture through it.
Okay, so kind of like filtering, but with a more specialized column.
Exactly. And then you have HPLC, or high-performance liquid chromatography, which is a more advanced version, using high pressure to speed things up and give you even more control over the separation. There are lots of other variations too, each designed to separate specific types of molecules.
Got it. So chromatography is like a whole family of techniques, each with its own specialty.
Exactly.
Okay, so we've got our three core methods. Crystallization to get those pure solids, filtration to remove solids, and chromatography to separate based on those unique molecular interactions.
Those are the heavy hitters.
Now, I think it's clear why getting rid of unwanted stuff is important. But I'm curious, why is achieving this super high level of purity absolutely vital for the person taking the medication? Why can't there be even a little bit of something else in there?
That's a really important question. High purity is non-negotiable because it directly impacts how safe and effective a medicine is going to be. You want to make absolutely sure that people are getting just the drug molecule, not any sneaky impurities or byproducts that might cause problems.
So it's not just about having the right active ingredient. It's about having only that ingredient and nothing else that could mess things up.
Precisely. Even tiny amounts of these unwanted substances could cause unexpected side effects or stop the drug from working properly.
So it's kind of like making sure there are no unwelcome guests crashing the party in your body.
Exactly. You want the drug to be the star of the show and no one else messing with its performance.
Makes sense. So it's all about maximizing effectiveness and minimizing any potential risks. Now, I imagine achieving this pristine level of purity is a huge challenge.
Hmm.
How do scientists even begin to figure out which of these methods, or maybe even a combination of them, is the best way to go?
Yeah, it's not a one-size-fits-all situation. The choice really depends on the specific drug and the impurities that are hanging around. It's like a puzzle where you have to find the right pieces and fit them together.
So it's a lot of detective work, understanding the properties of both the good stuff and the bad stuff, and then finding the best way to separate them.
You got it. Scientists have to carefully consider the physical and chemical characteristics of both the desired compound and the unwanted materials to choose the best approach. There's no magic bullet that works for everything.
Right, so each drug purification is its own unique challenge. And I remember reading that solubility, how well something dissolves, plays a huge role in all of this. Why is that so important?
Solubility is crucial, especially when it comes to crystallization and chromatography. For crystallization to work, you need your drug substance to be dissolved in a solvent first. That way, you can adjust the conditions to encourage it to crystallize out in its pure form.
So you need to find a solvent where the drug is happy to dissolve under certain conditions.
Exactly. It's like finding the right dance partner for your drug molecule.
Okay, got it.
And the solubility of the impurities is equally important. You want them to either stay dissolved or behave very differently so they don't crash the crystallization party and contaminate your pure drug crystals.
So it's all about finding that sweet spot where your drug is soluble under some conditions, but the impurities aren't, so you can separate them.
Exactly. It's a delicate balancing act. And in chromatography, solubility in the mobile phase is essential for those molecules to travel through the system and get separated. How well they dissolve also affects how they interact with the stationary phase, which leads to their separation.
So solubility is like a key factor determining how the molecules move and interact throughout these purification processes.
You could say that.
The research also mentioned that an ideal drug substance should have a certain minimum aqueous solubility. What does that even mean? And why is that benchmark so important?
That's a great question. When they talk about aqueous solubility, they're referring to how well a substance dissolves in water.
Oh, right, because water is kind of the basis of everything in our bodies.
Exactly. And that guideline of a minimum aqueous solubility, often around 5 milligrams per milliliter, but ideally over 50, is super important for how well a drug is going to be absorbed into your body.
Okay, so this is about making sure the drug can actually get into our system and do its job.
Absolutely. For a drug that's taken orally, like a pill you swallow, it needs to dissolve in the fluids in your digestive system before it can be absorbed into your bloodstream.
So it's like, if it can't dissolve, it can't get where it needs to go.
That's a good way to put it. It's like trying to send a letter without enough postage. If a drug can't dissolve properly, it might never reach its target in your body to have its effect.
That makes total sense.
Yeah.
Now, the sources also talked about something called the solid-state properties of a drug, including things like polymorphs and amorphous forms. I'm not quite sure I follow that. How do these different solid forms impact purification?
Okay, so this gets a little bit more into the molecular level. Basically, the specific solid form a drug takes can really change its solubility and how fast it dissolves.
So it's not just about what the molecule is, but how it's arranged in its solid form.
Exactly. It's like having the same Lego bricks, but arranging them in different ways, you end up with different structures that have different properties.
Ah, okay, I see.
So, for instance, an amorphous form, which doesn't have that nice ordered crystal structure, tends to be more soluble compared to a crystalline form.
So an amorphous drug might get into the bloodstream more easily.
It often does, but there's a trade-off. Amorphous forms can be a bit less stable and might change over time into those more stable but less soluble crystalline forms.
Ah, so like it might dissolve quickly, but then change its structure and become less effective over time.
That's the challenge. And then you have polymorphs, which are different crystalline forms of the same drug. They can also have different solubilities, which can affect purification and how they behave in the body.
So it's a whole balancing act between finding the right form that dissolves well enough to be absorbed, but is also stable enough to last.
Exactly.
And of course, scientists need to be able to control which form they end up with during purification and manufacturing.
Right.
Okay, so we've talked a lot about achieving this high level of purity. But what about the practical side of things? How do scientists ensure they have enough of the purified drug to meet the needs of all the patients who might need it? How do you scale up from the lab to the factory?
That's a crucial question. A chemist in a lab might be able to purify a tiny amount of a substance really well. But when you need to produce kilograms or even tons of a drug, things get much more complicated.
Right, because what works on a small scale might not translate directly to a huge industrial production line.
Exactly. Things like mixing, temperature control, and even the way you start the crystallization process can behave very differently when you go from a small flask to a giant reactor.
So it's not just about multiplying the recipe, it's about rethinking the entire process.
Absolutely. Maintaining that high level of purity and getting a good yield at such a large scale is much more complex. That's where process development comes in. It's all about fine-tuning and optimizing each step of the purification process to ensure you get both high purity and a decent amount of drug reliably and cost-effectively.
So it's not just about science, it's also about engineering and efficiency.
Exactly. It's about finding that sweet spot where science meets practicality.
You mentioned that you had some real-world examples from the scientific literature.
Oh, yeah.
Can you share some specific cases of how scientists tackle these purification challenges in pharmaceutical research? You know, I'm always fascinated by those behind-the-scenes stories.
I love those too. Well, the journal OPR&D, or Organic Process Research and Development, is a great place to find those kinds of stories.
That's good.
Let's look at a few that show different purification approaches in action.
Great.
So, in one study, researchers were working on an efficient way to synthesize a specific type of molecule called an amino acid. They used a cool process called de-racemization. Imagine you have a pair of gloves, right?
Yeah, a left and a right.
Right. Well, some drug molecules exist in two forms that are mirror images of each other, kind of like your left and right hand. Often, only one of those forms is biologically active, the one that fits the target in your body.
So it's like trying to fit a left-handed glove on your right hand. It just doesn't work.
Exactly. So de-racemization is like a clever way to convert the wrong-handed glove into the right-handed one.
I see. So they're essentially converting the inactive form of the molecule into the active form.
You got it. In this case, they oxidized one form and then chemically converted it into the desired form.
So they're manipulating the molecule to get the right version.
Exactly. And the result. They managed to isolate the pure desired enantiomer with a yield of 66% and an incredible purity of 99.9%. That means they pretty much got only the right-handed glove with almost none of the left-handed one.
That's impressive.
They even tested a combined approach using both chemical and enzymatic steps and achieved similarly great results.
So using nature's tools, enzymes, to refine the process even further.
Exactly.
What about another example where purification played a really key role? What other challenges did you find in your research?
Here's another interesting one that involved purifying an intermediate compound in the synthesis of a drug called tyranobant.
Okay, tyranobant.
One of the reaction steps used a palladium catalyst to speed things up. But the tricky part was that this catalyst had to be removed to super low levels in the final product.
Ah, so it was helpful during the process but couldn't stay in the final product.
Right. And you know what they found? A simple filtration step using activated carbon, a specific type called Darko-KBB, was really effective at getting rid of a pesky phosphine oxide impurity and bringing down the levels of that palladium catalyst to well below the acceptable limit.
So sometimes a relatively simple solution can solve a complex purification problem.
Absolutely. And what's even cooler is that this filtration step not only purified the compound, but also simplified the entire process. They were able to skip a more complex aqueous workup procedure. Less steps means less time and less cost, which is always good.
Efficiency and elegance, always a winning combination.
I agree. There's another example that beautifully shows the power of forming a salt of the drug molecule and then using crystallization. In the synthesis of nafoxidane, a key step involved converting the molecule into a specific hydrochloride salt.
So they're temporarily tweaking the molecule's chemistry to make it easier to purify.
Exactly. Forming a salt can really change the solubility and how a drug crystallizes.
Ah, I see.
And by carefully controlling the crystallization process, they managed to isolate nafoxidine with a yield of 65.1% and 99% purity, which is pretty impressive.
That's a pretty neat trick.
It is. And, sometimes filtration isn't just about getting rid of those tiny chemical contaminants.
Oh. What else could it be used for?
Well, they use it to remove larger materials, too. There was a case involving the large-scale manufacturing of a chiral intermediate, a molecule with a specific 3D structure.
Okay,
They used an enzyme attached to a solid resin to help create the right structure.
I see.
But after the reaction was done, they needed to remove that bulky enzyme-resin complex.
Makes sense.
And you know what they did? Simple filtration. They filtered the mixture to remove the resin and then washed it to collect any leftover product that might have gotten stuck. This straightforward filtration step allowed them to isolate the chiral intermediate with very high purity.
So filtration can be a versatile tool, tackling impurities of all sizes.
It really is.
Now, before we wrap things up, any other interesting or maybe more modern purification techniques that you came across?
Oh, there are always new advancements happening. One cool example I found involved continuous flow purification.
Continuous flow. What's that?
It's kind of like an assembly line for chemical reactions. Instead of doing everything in batches, the reactions happen continuously in a flowing stream.
Oh, that's interesting.
In this case, they used a continuous flow process to synthesize a chiral compound using a reaction called the Shapiro reaction.
Okay.
And get this, once the reaction was done, they purified the product with a simple washing step and then filtered it through alumina. That's aluminum oxide, a common material used in purification.
So pretty straightforward steps.
Pretty much. And they ended up with an impressive 80% yield and 96% purity.
Wow. That's pretty remarkable for such a seemingly simple approach.
It is. It shows how continuous flow systems can sometimes make purification much more streamlined.
So sometimes innovation lies in simplifying things. Well, these examples have been so fascinating. It's amazing to see the range of techniques scientists use to purify drug substances, from using enzymes to manipulating molecular structures to good old-fashioned filtration. It really highlights the ingenuity and meticulousness that goes into making the medicines we rely on.
It really does.
So to recap our deep dive today, we explored the fundamental methods of purifying and isolating drug substances, those essential ingredients that help us feel better.
Yeah.
We talked about crystallization, filtration, and chromatography, and we saw how each of them plays a crucial role in getting those pure drug compounds that eventually end up in our medicine cabinets.
And we saw that achieving that high level of purity isn't a simple task. Pharmaceutical scientists and engineers have to be incredibly innovative and dedicated to scale up these processes and produce enough medication to meet the needs of everyone who might benefit from it.
Right, and as those real-world examples showed us, it's a constant process of refining and optimizing, finding that perfect balance between science, efficiency, and practicality. It's really amazing to see how much effort goes on behind the scenes to ensure the quality and the safety of the medicines we take. It makes you appreciate those little pills even more, doesn't it?
It really does.
So, next time you pick up your prescription, take a moment to think about that incredible journey from the lab to your hands. It's a fascinating story of science and innovation at its best. Thanks for joining us on the Deep Dive.
Thanks for having me. It's been fun.